A bit to get through today, so I'll go ahead and kick things off. Hello, everyone. I'm Casey from Cambridge University Press, and I would like to welcome you all to today's Quantitative Plant Biology webinar. For those of you who don't know, Quantitative Plant Biology is an open access journal co-published by Cambridge University Press and the John Ennis Center. We just celebrated our 5-year anniversary of publication, so if you haven't done so, please go and check out our website and some of the amazing research that's been published over at cambridge.org/QPB. I'll drop a couple of links for you all in the chat. Before I pass along to our speakers, I just wanted to cover a couple of quick housekeeping items. Those of you who are attending are in listen only mode, but we do want to encourage questions today. So if you have any questions, you have a couple of options. At the bottom of your screen, you should see two different buttons. There's one called Q&A and one called chat. You can put your questions in either of those places, and we'll endeavor to answer those during our Q&A session at the end of today's talk. And lastly, if you have any technical difficulties today, trust pop those into the chat function as well, and I or a member of our team will do our best to help you. But that's enough rattling from me. I will hand over to Ingo to introduce today's speakers. So thank you.
Yeah, thanks a lot for the introduction. Thanks to everyone for joining. I welcome you now officially to this webinar. So we are in the journal Quantitative Plant Biology is from John Ennis Center and Cambridge University Press, is a non-profit making publisher. And within this journal, we had then decided to organize a special paper collection about quantitative approaches to cellular aspects of plant homeostasis. For this, Dale Sanders, the former director of the John Ennis Center, and me, we joined forces, and we then had a look what can we do to bring this collection on the run. And we then took it and we thought, well, in plants, we have about 20 essential elements. And so we had to look at the periodic table. And within these elements, we could then identify about 12 elements where we could identify people who could contribute with their expertise in these different elements. So at the end, we collected 12 articles for our collection, each focusing on an element that is important for plants, mentioned here by circles. Additionally, we had 2 articles that shed a bit light on thermodynamics of homeostasis, homeostatic processes in plants. And today we could gather 6 speakers. The first speaker will be Edgar Peiter. He will talk about manganese handling in plants, advances in the mechanistic and functional understanding of transport pathways. Edgar is from the Martin Luther University in Halle-Wittenberg in Germany. Thereafter follows Ute Kremer from the Euro University in Bochum in Germany. And she will focus on zinc and talk about changing paradigms for micronutrient zinc, a non-protein cofactor as a signal relaying also cellular rhotic state. Thereafter, we will switch over to Foshan University in China. Lars Henrik Weger from there will take care of potassium and their potassium homeostasis and signaling from the whole plant to the subcellular level. Thereafter, Colin Brownlee will provide his expertise about calcium. He is from the Marine Biological Association in the UK, and he will talk about cellular calcium homeostasis and regulation of its dynamic perturbation. Following there is then we switch over on the right to the periodic table with chloride. There, Christoph Martin Geifus from the Geisenheim University in Germany and Stefanie Wege from the University of Bonn in Germany. They will present something about chloride transport and homeostasis in plants. And last but not least, we switch to the metal copper. There, Olena Vatamaniuk from the Cornell University in the United States will talk about copper connections, coordinating transport, sensing, and systemic signaling in plants. So this is the overview of our webinar today. Now, I would like to take the opportunity to announce already something which will happen in 2028. We have an international workshop on plant membrane biology, and this time it will be the 21st International Workshop in Plant Membrane Biology. It will take place between the 27th of March and the 21st of March. 2028 is just 2 weeks before Easter, so you may combine it with Easter holidays. And it's a great pleasure for me because after 60 years of this conference in all parts of the world, in all different continents, it is now for the first time in South America, and it will be Chile hosting this time the workshop. So please save the date. Chile is a lovely place to visit, and probably you can combine it with Easter holidays. Well, with this, I would go now, hand over to the collection, hand over to the speakers, and hopefully this webinar will also raise a bit your curiosity to have a closer look to other articles also in this collection. So with this, I now stop and I will hand over to Edgar, and Edgar will now talk about manganese handling in plants.
Thank you very much, Ingo, for the nice introduction. And I now try in 5 minutes to get you interested in manganese. Really exciting element. So if we start with the next slide, please. Yes. So being located in an agricultural university, I have to show you a field. And this is a crop that is severely manganese deficient and it's chlorotic. And next, please. The reason is that this soil is alkaline and well aerated, and this renders manganese unavailable. Manganese is taken up as manganese 2+, and it's then oxidized into insoluble manganese oxides. Yes, next, please. The reason why these plants are chlorotic, yellow, is for manganese function in photosynthesis. It's a main constituent of photosystem 2. Next, please. Where it is located in the oxygen evolving complex. Next, please. And in the oxygen, we have four manganese coordinated with calcium and oxygen as well. And the manganese takes the electron from the water, thereby leading to the splitting of water. And with also, of course, oxygen and protons being left. So the water splitting, oxygen evolving complex is a primary function of manganese in plants. And next, please. It's not the only one in photosynthesis. Next, please. Another function lies in the detoxification of reactive oxygen species in the mitochondrion, for example, in the superoxide dismutase that contains manganese. A third prime function, next please, lies in yet another organelle in the Golgi apparatus, where manganese is absolutely required, essentially required for the transfer of sugar residues, either to proteins in protein glycosylation or for the synthesis of cell wall matrix polysaccharides. And all of these enzymes that are indicated here, they require manganese. So this brings us to the topic of the review. Next, please. So yes, how does actually manganese reach its targets? So being taken up from the soil and then passed on right to the organelle. There are several steps that are, of course, of importance. The first one, next please, being, of course, the uptake pathways. Then we have a transfer into the root central cylinder, interactions possibly in the vascular system. Next, please. And where manganese might be also stored and allocated differentially to different organs and different leaves. Next, please. And within the cell, then of course we have a distribution to different organelles. Yes, next please. For these processes, the plant, of course, has a toolbox of manganese transport proteins that are all assembled here in a virtual cell that contains our current understanding of manganese transport. And if we zoom in into a few of them, so if you go, yes, next please. So uptake, we know for some time now, is mediated by proteins of the NRAM family and also as a byproduct by an iron transporter, IRT1. Next, please. Then after transfer into the leaf cell, we are known now since a few years the manganese transporters in the chloroplast here called bicat-2 to load the chloroplast and bicat-1 to pass it on into the thylakoid for the feeding of the photosystem 2. Yes, next please. The same family actually loads the Golgi with manganese to feed processes involved in glycosylation that I've shown you before. And next please. The vacuole on the other hand, is loaded by another family of MTPs under specific conditions only. It can be unloaded yet by transporters that belong also to the NRAM family that are also involved in the uptake of manganese. Next, please. Yet another NRAM transporter has been shown to actually unload manganese from vesicles belonging to the Golgi network after the Golgi, and this process is supposed to play a role in cellular manganese homeostasis in feeding the cytosol again with manganese from these organelles, then to be taken up by bicat-1. Yeah, so this gives you a little impression of manganese pathways on plant and organellar level. And next, please. A main question that remains is, of course, how are these transporters regulated? If they all run on full speed, the plant would collapse. So there are, of course, different layers of regulation. And next, please. Comes quite often as the first thing, transcriptional regulation. That's what everybody measures. In the case of manganese, this is really the exception. Most transporters are not regulated on transcription level, with the exception maybe of MTP8, that is strongly induced by iron deficiency, for example. Yes, next, please. Another layer is the processing of RNAs, and we recently have uncovered, there was a postdoc in my lab, Santiago Alejandro, who did this project, that there's alternative splicing for manganese transporters. And next, please. And this alternative splicing can have two effects. One effect might be a change in substrate. So by alternative splicing, we either have the capability to transport iron or not. So this is one layer of regulation. And next, please. Another manganese transporter, alternative splicing, led to a change in localization of the protein. What we are not aware of yet, what we don't know yet, is the physiological relevance of these changes. This is a really active area in the field. So if we stay with localization, we can actually go one layer up in protein regulation. If you show me the next, please. And this is the modification of the protein by phosphorylation, for example. And it's been shown in Cathy Curie's lab that the phosphorylation of the uptake transporter NRAM1 leads to clathrin-mediated endocytosis, in particular under high manganese conditions. And this is to protect the plant to take up too much manganese, and so the transporter is just internalized. Yes, next, please. A similar regulation by phosphorylation can not only change the location, but also the function, the functionality of the transporter. So it's been shown that, again, the uptake transporter NRAM1, the activity is regulated by phosphorylation. And this phosphorylation is actually brought about by calcium-dependent kinases. And now I'm coming towards the end, or I'm over time already. So it's calcium dependent, these kinases, and this fits very well to a finding also in that story from the Kudla lab in Münster. Next, please. That under manganese deficiency, there's an induction of long-lasting, very slow calcium oscillations originating from a bunch of cells that are not defined otherwise. So it's believed that this calcium signal translates into the activation of NRAMP1. You've seen that there are well, pieces of the puzzle in different layers of regulations that seems to all kind of regulate manganese homeostasis. So with this, I'm coming to the end. Next, please. Indeed, what we have are pieces of the puzzle, and many questions are still open. Now, maybe you can flick just through the questions. So what we don't know is actually how is cellular manganese homeostasis integrated? How do organelles cooperate? So I've got one ion of manganese. Where does it go? To the Golgi chloroplast. How is this coordinated? Next, please. Then going up one level, how is manganese allocated on whole plant level? So we've got some ideas, especially from cereals, that in the node, for example, there can be like a switch to old and young leaf, but there are really gaps in our understanding in this. And finally, manganese is not alone. How does it interact with other cations that might bind to the same sites that are transported by the same transporters? Yes, there is specificity. So all in all, finally, next, please. We have a lack of quantitative data, and I would like to invite everybody to join the manganese field to get there. Yes, thank you very much.
Yeah, thank you, Edgar. So we immediately switch over to another metal is zinc, and we switch to Ute Kremer from Bochum.
Hello. I describe in this article and develop a framework of how plants use and handle zinc. And I develop a few quantitative considerations that are partly coming from other organisms and partly from plants to move these concepts a bit further, although experimentally we don't know much about it in plants yet. I also go into describing arising concepts and also terminology that we need to use in plants in order to correctly describe the homeostasis and handling of zinc and of other metals. And what I want to show here is just a few basic things on which this article is built, just to give you a taste how we must shift our concept of metal homeostasis in general. So can we go to the next slide? So zinc deficiency, just to begin with the topic of this article, is a serious problem. A large number of people, 2 billion people worldwide, are thought to be zinc deficient. And this affects primarily the health of children, in fact, although zinc deficiency is widespread in all types of population groups. And up to 50% of the soil that is used to cultivate cereal crops is zinc deficient. And these two images taken from another review article show how well the zinc deficient population matches the areas with zinc deficient soils, really highlighting, the next please, how important plants are in governing human zinc status. So this table of the elements, periodic table, which you've seen before, highlights mineral nutrients we all need and also plant needs, and they are divided up into macronutrients needed at somewhat higher concentrations than micronutrients, with the macronutrients in plants highlighted here in green and the micronutrients in yellow, and you see zinc down there in the corner with the zinc word written over it. And can I have the next, please? So what is special about zinc and all these chemically similar metals that are in its vicinity, like manganese that was just spoken about, iron, nickel, and copper, they all have highly similar chemical properties, and they differ fundamentally in their chemistry as cations from what we normally conceptualize as a cation. We conceptualize cations mostly based on what we know about sodium and potassium cations that are mostly present as free cations in solution and don't interact much and don't interact long-term with any electrostatically bound molecules. However, zinc establishes quite stable so-called coordinative bonds that have a nature in between these electrostatic bonds and atomic bonds that we typically know from carbon-carbon bonds. Zinc is in between with the bonds it establishes, and it likes to share electron pairs with several donor atoms, preferably of the same molecule, like a protein, and thus defines in a geometrically defined coordination sphere its interactions with molecules, which is broadly used in biology. Next, please. So there are thought to be around about 2,000 zinc-dependent metalloproteins encoded in the Arabidopsis genome, where zinc either acts to form the three-dimensional structure of the protein or acts as a catalytic cofactor interacting and modifying substrates. Zinc is special among all the metals in its vicinity by being present in relatively high concentrations inside cells, but also having a comparably high affinity for thiols. And a typical so-called complex that zinc likes to form and will form abundantly, for example, in the cytosol, is a zinc bis-glutathionato complex, as you can see in the next, please. The next one, so a zinc glutathione complex. Glutathione is present in high levels in the cytosol, and this complex is going to be abundantly present. Next slide, please. This represents some kind of a thought experiment that people have been asking themselves for a long time. How do nutrient metal ions reach their correct metal binding site? On the right, you see something that symbolizes a cell with a number of so-called apometalloproteins that need to bind metal specifically to exert their specific activity. And the needed metal ions are symbolized by different shapes and colors on the left side. When you now imagine you take a mix of all these metal cations in excess and pour it over the cytosol, then what you get out of it is, next please, that copper actually ends up bound to every single protein. And this is not what you want, because it will kill the cell. And ultimately, this is reflected, next please, by the so-called Irving-Williams series on the order of decreasing binding affinities. And copper binds to all kinds of ligands with the highest affinity. And zinc you see coming pretty close to copper, and magnesium and calcium you see right at the bottom there. So magnesium and calcium binding sites, when these other metals are present, are going to be occupied by zinc, for example, and magnesium and calcium can also be displaced from their binding sites by these cations, meaning that cells need to control them at all times in order to prevent that. Next, please. In addition, this means, and I think the thought experiment shows you, that a diffusion-based model of how metals reach their binding sites and their apometalloproteins is unfeasible. And people have thought about it for a long time. And the first model that was developed for this, next please, is a so-called paradigm of metallochaperone-mediated cellular protein methylation, and Olena will know this very well. This is a yeast cell, and this is the idea that copper enters cells through a copper transporter, shown on the left, called CTR, and then so-called, next please, metallochaperone proteins take over the copper by protein-protein interaction. The copper is transferred into this metallochaperone protein. Then the metallochaperone protein goes to the target protein and via protein-protein interaction donates the metal cation in a targeted manner to specific apometalloproteins. Now, quite a few metallochaperone proteins were identified for copper, meaning that this model is feasible. And people assumed when this was discovered by Tom O'Halloran and Valeria Colotta that this was happening for all metals. But in fact, for other metals, and especially in plants, we found almost no metallochaperones. Next, please. In addition to the metallochaperones, so-called metallothionein or metal-binding proteins that offer as some kind of metal sponge to bind excess metal were also thought to be important in that model. And ultimately, next please, it was shown by them based on the affinity of cellular sensors that trigger detoxification responses. It was estimated that there is less than a single free copper ion in the cell. All copper is bound in the cell, and the concentration of free copper then comes down to less than 1 attomolar. Further developing this concept, next please, the so-called paradigm of cytosolic buffer-mediated cellular protein methylation was developed, and the group of Nigel Robinson had an important role there. They expanded this concept and they quantified in vitro the metal binding affinity of a large number of bacterial metal sensor proteins that bind the metal and then trigger transcriptional responses to metals. And the diagram on the left shows these affinities, and the highest affinities of these metal sensors is found for copper. And this means that when copper concentration goes above this level, a response is triggered. And they took these affinities as a proxy of the cellular metal concentrations. And what you see here with the lowest copper then going up zinc, cobalt, iron, and manganese is, next please, that the available metal concentrations inside the cytosol of bacterial cells are actually the inverse of the Irving-Williams series. So when you think about it, it is actually quite logical that this might happen in cells, but this has actually a major consequence of how metals can be integrated into the proteins, and it means that they can be integrated into proteins from the cytosolic cellular buffer consisting of all metal binding sites, including no molecular mass compounds, in particular, for example, also glutathione. Next, please. So this model for sure is biologically relevant for the transfer of metals into proteins. And actually, when you think about it, especially for zinc, it has implications for the homeostasis of all metal ions. So the homeostasis of all metal ions is tightly interconnected, and excess of one of them will affect the homeostasis of all others. In addition, by modifying the concentrations of free thiols inside cells through redox stage transitions, the metal availability will change in cells, and in particular, the availability of zinc will change in cells. This is also predicted to happen when cytosolic pH changes, because that will also change zinc binding inside cells. And from this, there comes the idea, which has been explored a little bit already in mammalian cells, that zinc cations actually have a role in controlling cellular signaling and regulation through facultative zinc binding sites that are just regulatory. Now, in plants, we know virtually none of this. Can we move on, please? There are only a few examples of regulatory zinc binding sites known in plants, and there's really a need for quantitative biological experiments addressing these novel paradigms in plants, and especially also addressing what follows from these. Thank you.
Thank you, Ute. Now we are moving quickly to alkali metals, so this time potassium. And I hand over to Lars for
Yes, I hope you can hear me. So potassium is a macronutrient, and it's the most abundant cation in plants, can take up to 10% of dry weight. And there's a bulk of literature on all aspects of K, particularly on K transporters. So there are 75 transporters in plants which can transport K, and 35 of them are highly K selective. But we didn't want to be repetitive to previous reviews on this aspect. So we wanted rather to focus on aspects of K which are somewhat underrated so far. We think they deserve more attention. And so one of these aspects is circulation of K at the whole plant level. Then we come to a modeling approach of K across the plasma membrane of plant cells. Then we want to point out that K is not just an osmoticum, but can be a messenger and an important signaling element in plants. And finally, we will talk about vacuolar K and K transport across the tonoplast. So next, please. Next, please. Yeah. So we start with K circulation in the plant. So K is constantly on the move. It moves upwards in the xylem with the transpiration stream, and then part of the K is unloaded there in the leaf. But a large part actually is loaded into the phloem and then moves back to the root. And there are several sites of regulation in this whole plant pathway. One is, of course, K loading into the xylem by xylem parenchyma cells, and the main player is SCORE, a stellar K outward rectifier, which is regulated by the K gradient across the membrane. And of course, here K is also interacting with the transpiration stream, so higher volume flow would also accelerate K loading into the xylem. Now, another site of regulation is phloem loading, and here AKT 2 or 3 is the most important. And this transporter can, this channel can act in two modes, in a voltage-dependent mode, where it actually serves K loading into the phloem, and a voltage-independent mode, where it clamps the membrane potential of the sieve tubes to a K. So it serves then as a driving force for assimilate uptake by phloem. Now, it's an obvious idea that the amount of K, back please. It's an obvious idea that K traveling from the shoot to the root can serve as a signal on the K status of the shoot. And indeed, there is good evidence from split root experiments showing that. And the idea is that the stellar apoplastic K concentration is affected by phloem unloading, and this in turn will regulate SCORE activity and regulate K loading into the xylem vessels. Okay, next please. Yeah, now I refer to the great work of Ingo. Probably he could better report on this than me. It's about modeling of K transport across plant membranes. And the idea is to use a very basic level of modeling, just leaving aside all the details of regulation and just turning to the key functions of these transporters. And on the right side, you see what they call the K homeostat. And it consists of a set of 3 transporters. So you need a proton pump, you need an HK antiporter, and you need a K channel. And these 3 transporters are required to keep membrane homeostasis, so to keep a constant K gradient across the membrane. So you really need for that purpose metabolic energy, so the proton pump. And you have futile cycling of both protons and K across the membrane. And on the left side, you see that, proton efflux and K influx via the channel must be of the same magnitude for electron neutrality. And this basic model has been extended now, also including perturbations of the membrane, for example, an action potential, and all this, or much of this actually has been published in this journal. Okay, so we go on to the next one, please. Next slide. Yeah, it's generally still believed that K is just a boring osmoticum, but this is in fact not true. It has emerged in recent years that it can serve as a very important signaling molecule in plants. And some of these signaling functions are listed here. So first of all, what is called metabolic switch. So many enzymes of glycolysis or TCA cycle depend critically on the K concentration. So when K goes down in the cytosol, for example, under salinity stress, then these enzymes will be downregulated. So that would downregulate anabolic reactions and favor defense mechanisms in the plant. So the K can really reshape the whole metabolism of plants. Another important aspect is that K deprivation favors autophagy and programmed cell death. And on the other hand, high K concentration would protect the plant cell against these mechanisms. And finally, it turns out that the dynamics of K, so the patterns of K with time, themselves can contain information similar to what is known, for example, calcium. And potential decoders of this information are, for example, the protein ATPases, which are regulated by K. So K favors inactivation by dephosphorylation, or the formation of cyclo AMP, for example, from ATP, is also strongly dependent on K. Okay, next slide, please. Finally, we turn to the vacuole as a K reservoir. So it is known that the K level in the cytosol is quite tightly regulated and kept on a more or less standard, constant value, even under K deprivation. And this buffering function is brought about by the vacuole. And we have a number of transporters in the tonoplast whose interaction is not fully understood yet. For example, we have the TPC, or slow vacuolar channels, or also fast vacuolar channels. And it's now believed that they mainly serve as a security valve when the vacuolar K concentration goes up. But on the other hand, when vacuolar K is low, then they would prevent a backflow of K from the cytosol to the vacuole. And another important player here is our TPK channels, K-selective voltage-independent channels that provide K to the cytosol under high cytosolic calcium concentrations, for example, under salinity stress. So these are some aspects of K that I would like to wanted to highlight here, and I pass to the next element.
Yeah, thank you, Lars. I pass over to Colin Brownlee, who will now go on with calcium.
Okay, thank you. So I'm going to address cytosolic calcium largely in this short presentation, and its homeostasis and how its perturbation from that homeostasis can be regulated, and why it does that. So we can consider cytosolic calcium, which is kept very low in the cytosol, around about between 100 and 200 nanomolar in resting cells. You can consider that to be maintained by the balance of calcium influx mechanisms and calcium efflux mechanisms, where calcium influx will cause an increase in cytosolic calcium and calcium efflux will cause a decrease. And the shape of those elevations of calcium can be modulated by the presence of calcium buffers. So the article addresses various aspects of those calcium influx and calcium efflux mechanisms. On the bottom right-hand side of the slide, you can see the various compartments that are proposed to be involved in maintaining the calcium homeostasis and, in fact, determining the shape of certain calcium elevations, which we know in all eukaryotic cells form the basis of calcium signaling via different calcium signatures, so different patterns of spatiotemporal elevations of cytosolic calcium. So these compartments include the apoplast on the outside of the cell, the vacuole, the endoplasmic reticulum, the chloroplast, and the mitochondrion. And I've also included some mention of the nucleus here as an important self-contained signaling compartment in the eukaryotic cell. The red arrows here represent efflux into the cytosol, or influx, sorry, into the cytosol from various compartments, and the green arrows represent efflux from the cytosol into these compartments. So one of the major questions is, what is the relative role of these different compartments in determining calcium homeostasis and in determining its perturbation, which is the fundamental basis of calcium signaling? So next slide, please. So one thing we have to do to be able to do that is to be able to quantify the different driving forces and concentrations in these different compartments. So I've represented by these blue triangles the direction of the electrochemical potential driving force for calcium into the cytosol or out from the cytosol. And we can see from the apoplast, because of the very negative membrane potential, we have a large inward driving force for calcium. And similarly, from the vacuole, there is a large driving force for calcium to enter the cytosol. The endoplasmic reticulum also, because of its higher concentration than the cytosol, despite it not having a significant membrane potential, potentially can act as a source of calcium for influx into the cytosol. I've also included the mitochondria and the chloroplast, and because of their membrane potentials, they actually can act as sinks of calcium, whereby calcium will tend to move into these organella compartments from the cytosol. Okay, next slide, please. If we consider plasma membrane calcium entry channels, I've just listed some examples of the calcium entry channels that we are familiar with in plant cells. I'm not going to go into any details in listing these. They include cyclic nucleotide-gated channels, glutamate receptors, mechanosensitive channels, nucleotide-binding leucine-rich repeat receptors, and these have all been characterized to various degrees at the molecular level. We also know that there are depolarization-activated calcium channels and hyperpolarization-activated calcium channels, which have been characterized physiologically, but to date there is no their molecular components haven't been determined. But one point I want to make with this slide is that when calcium enters the cytosol from the apoplast or from the external medium through calcium channels, it doesn't actually get very far. And the little figure on the bottom right-hand side illustrates this. And this is just from some work that we did several years ago with the rhizoid of the brown algal cell, fucus. And using confocal line scan imaging, we were able to detect very discrete and very transient elevations of calcium, which we proposed were due to a channel opening and then closing and then opening again. The point I want to make here is that this doesn't actually have a huge impact on the cytosolic calcium beyond the channel mouth. And so we have to agree that plasma membrane calcium influx is augmented by release from intracellular stores, except for these highly localized calcium elevations. And there are several examples in the literature now from plant cells, for example, in the venous flytrap, whereby calcium entry through a channel is actually augmented in terms of elevating cytosolic calcium by a release of calcium from intracellular stores. Okay, next slide, please. I won't go into any details on this table. This is presented in the article, so if you want to know more about it, please have a look at the article. I'll just summarize on the left some key points from this table, which is really summarizing the intracellular calcium sources and sinks and some of the transporters and channels that are involved in that dynamic regulation of cytosolic calcium. So first of all, we find that there are calcium uptake transporters on both vacuolar and endoplasmic reticular membranes, in addition, of course, to the ATPase transporters that are present on the plasma membrane. Secondly, there are calcium release channels on the vacuolar membrane that have been characterized, at least physiologically. To date, no plant endoplasmic calcium release channel has been described except on the nuclear envelope and in certain algae. And finally, chloroplast and mitochondrial transporters facilitate organelle uptake of calcium from the cytosol, and they can regulate their calcium independently. Okay, next slide, please. So finally, in terms of the outlook for the future, I've just posed some key questions here. And the first question is, what are the relative roles of plasma membrane and endomembrane calcium fluxes in different signaling systems? Secondly, we know very little about the nature of endomembrane release mechanisms in plant cells compared to what we know in animal cells. For example, is there any evidence at a molecular level of how calcium-induced calcium release might operate in a plant cell? The next point is that we know very little about the interactions between different endomembrane compartments. For example, the endoplasmic reticulum, the mitochondrion, and the chloroplast. In animal cells, it's well known that release of calcium from endoplasmic reticulum stores leads to an elevation of cytosolic calcium in very localized hotspots, which are close to mitochondria. And the mitochondria play a key role in bringing cytosolic calcium levels back down to resting. We have no evidence that these close contact points between endoplasmic reticulum and mitochondria have function or even exist in plant cells. And finally, another major question relating to plants is whether there is any store-operated calcium entry mechanism in the absence of any animal homologs characterized at a molecular level. And one example of that is the inositol trisphosphate receptor, which in the presence of ORI channels and STIM molecules, which together form complexes which, when intracellular endoplasmic reticulum stores become depleted, can lead to the influx of calcium almost directly from calcium channels in the plasma membrane into the endoplasmic reticulum that can top up the endoplasmic reticulum stores. There is evidence that depletion of ER stores in plants can cause defective calcium signaling, but to date there is no evidence at a molecular level for the machinery that can bring about store-operated calcium entry into plant cells. So on the right-hand side, I've just summarized some of the obvious approaches and technological advances that will allow us to address some of these questions in the future, which include molecular genetic characterization, the continued development of genetically encoded reporters, which has really seen amazing results in recent years. The continuing application of electrophysiology, and I can't stress that more strongly. We really do need to keep our electrophysiological expertise topped up as a community. And then great advances in subcellular and whole plant imaging with the whole range of different microscopies that are now available. And finally, in the article, I do address the role of different types of modeling in order to understand the basis of calcium homeostasis in plant cells more generally. So at that, I'll finish, and thank you very much.
Thank you, Colin. After now 4 cations, we move over now, we change polarity and we switch to chloride as an anion. And I hand over to Christoph Martin or to Stephanie, who is there.
Yeah, hi, I will start. Yeah, thanks for introduction. So in addition to Stephanie Wege and me, Ob Rolfsummer also contributed to the review, but he is unavailable today due to teaching duties. So the topic we reviewed is chloride in plants. And so chloride is a nutrient with 3 functional zones. So it's essential at extremely low concentrations, but beneficial at a macronutrient-like concentration range, and of course toxic when being taken up excessively. So next slide, please. So at a low concentration, plants suffer from chloride deficiency because it limits photosynthesis because it's required for the oxygen evolving complex in photosystem 2. But what makes chloride remarkable is that its beneficial zones spans surprisingly large, a large concentration range, much broader than that of most other micronutrients. So in this beneficial zone, it acts as a major osmoticum, maintaining turgor pressure and supporting water balance, of course. And it is thought that chloride is especially efficient because unlike other osmolytes, such as proline or malate, it does not require a costly metabolic synthesis. And of course, it's a higher concentration where chloride is mainly taken up together with sodium, it turns toxic. And very interestingly, all the plants differ widely in their chloride requirements. And having this said, I would like to pass to Stephanie Wege.
Yeah, thank you, Christoph Martin. Yes, so as Christoph Martin said, I'd like to focus now on what I speak about a little bit on this middle range. And I put this little schematic down there. It's not that scientific, so it's mainly to highlight, as already said, that plants do have a different requirement or preference for different chloride concentration. That's externally, but also internally. But I hope you all see there is a plateau where there is optimal growth, and this is over a wide range of chloride concentration. And next, please. Yes, so it's an osmoticum, so I won't speak a lot about that, but I want to focus on maybe what the other beneficial functions are. And this is more, I'm sharing my thoughts with you here a little bit, because I think this is an area where we should really focus a little bit more our research on. So next slide, please. Oh, maybe next animation as well. Something went a bit funky here, I think. Yes, thank you. So as I said, as you can see here, point 1, so chloride can function as a cheap osmoticum. So I want to shift directly to point 2, and I'm very glad Lars already introduced a little bit the homeostats. You can see on the right side, I also put a little schematic here. It's not from my publication, but this is these wonderful figures that are very nicely explained typically by Ingo and in his publications. And you can see on the left here in the schematic, the potassium homeostats that are really important for maintaining membrane potential, but also for adjusting cytosolic ion concentrations. And on the right side, you can see we have something similar for anions here. And I think that's probably where the chloride can come into play, also in higher concentration and not as this essential micronutrient in photosynthesis. So I'm just sharing my thoughts. I think chloride and potassium, you have these homeostats, and probably they allow for an optimal flexibility now for the cell and the plant to adjust ion concentrations transport at the membranes. And then in the last point, maybe there's another animation coming. Yes. So I think a lot of people, when they think about chloride, we think automatically on this antagonism with nitrate. And I would like to say, yes, I think at very high concentrations of chloride, this is happening. But what is in this middle range? Maybe there is, instead of an antagonism, maybe there is a synergistic effect here, because something what I always found a little bit puzzling for the chloride is, you can see here, I put the data from this quite old publication, that plants that are starved for both nitrate and chloride, they take up the chloride faster than the nitrate. So, and I think there's maybe something here that we're missing out. So is this maybe a preparation for the subsequent nitrate uptake? So you get this efflux and efflux of the anion happening at the plasma membrane and at the vacuolar membrane to then prepare the cell to quickly shovel the nitrate into the vacuole to store it there, and also to have the chloride for this influx and efflux to not lose valuable nitrate here. But of course, at high concentrations, it becomes toxic. And with this, I'd like to give back to Christoph Martin.
Yeah, thanks. Next slide, please. So this slide is about chlorine toxicity, and we try to illustrate what happens in the plant. So at the upper left side, you can see that when chloride accumulates in the apoplast, there's a competition between phosphate and nitrate uptake. So in general, literature reports about decreasing concentration of phosphate and nitrate when chloride is taken up excessively. And this is, of course, the first problem in terms of plant nutrition. And chloride is also, it seems to accumulate in the chloroplast and in the mitochondria. And what we know is that this is associated with a lot of reactive oxygen species that certainly cause damage. It is also a very old hypothesis that if concentration in the cytosol increase, that there is a problem with enzyme functionality, because the salt concentration is getting too large. So there are probably many reasons why excessive chloride concentration in the cell are causing problems, but there are also many, many open questions. And for instance, what are the energy costs of chloride cycling, if something like this exists at all? So how is chloride moved, and is it transported back to the root, being excreted to the soil? What about tissue specificity? Why is it accumulated in the root cortex when in the endodermis? So there are a lot of conflicting results from different studies. And we really do not know much about homeostasis. So how tightly is chloride homeostasis coupled to potassium, calcium, or manganese transport and homeostasis? So there are really many, many open fields on this topic. Yeah, next slide, please. Yeah, so we welcome you to team up with us, because even though chloride is only an ultra trace element, there are many, many open questions. Thank you for your attention.
Yeah, thank you, Christoph Martin, and thank you, Stephanie. Now we switch back to metals, real metals, and we have now copper, and Olena Vatamaniuk will talk about copper connections in the plant.
Well, hello, everyone. Well, first of all, thank you, Dale and Ingo, for inviting us to contribute this review, and also to the webinar. Thank you all for staying to the end. So this review is a team effort from my group. And so if you please hit 4 times next, you will meet co-authors, Rujen, Tatiana, Hannah, and Jana. So why do we like copper? Please hit next. So copper, as you know, is a micronutrient, readily accepts and donates electrons, and so is a cofactor for enzymes which are involved in electron transfer reactions. And so copper participates in photosynthesis, respiration, oxidative stress, and scavenging, oxidative stress scavenging in all organisms. In plants, copper also plays a role in cell wall lignification, pathogen resistance, and reproduction. I would like to highlight that beyond its static role as an enzyme cofactor, the dynamic signaling role of the labile copper is increasingly recognized in animal systems. And so it was shown that copper is actually needed and is required for the kinase activities of kinases that are involved in cell proliferation, autophagy, etc. in animals. While the signaling function of copper in plants is way less explored, copper, please hit next, is essential for hormone signaling. For example, ethylene receptors contain copper ions, and copper is required for ethylene binding. Next, please. Copper can also act as a systemic signal in long-distance shoot-to-root communication of plant status. Next, please. However, copper is also toxic if it is accumulated in a ionic state in excess, which is unlikely. You've heard it from Uta, but still it can accumulate in a ionic state in excess, and so it can cause damage to cellular protein and also cause oxidative stress. So in our review, we focus on how plants regulate copper uptake, cellular trafficking, tissue distribution, storage, and utilization. We also explore the differences in copper uptake strategies between grasses and non-grass species, the proposed mechanisms of copper sensing, and imaging evidence for systemic copper signaling. So here I'll just provide you a taste of what is discussed in the review in quite detail. So please, next. Yes, so just some of the highlights for the copper transport processes in dicots and non-grass monocots. So copper in soils predominantly exists as copper-2 in oxidized form in aerated soils, and it forms complexes with organic matter, iron, and aluminum oxides. And so copper must be mobilized before uptake into the roots. Studies in Arabidopsis thaliana showed that copper-2 is reduced to copper-1 before uptake. And so Uta's lab have shown that members of the Fro family, Fro4, Fro5, are involved. Reduced copper is then taken up into root epidermal cells by members of the CTR-COPT family, and mainly COPT1 and COPT2 are the players there. Uptake of copper in the oxidized form as copper-2 via ZIP2 transporter was suspected for at least 2 decades, but only recently was demonstrated by the Barberon lab. There's a number of transporters which are involved in copper loading into the xylem in the root, reabsorption into the xylem parenchyma cells, lateral transport, loading into the phloem were identified. They are all listed here on the right, and they belong to HMA, YSL, OPT, and NAT families. They are discussed, and they're in this, uptake and transport strategies are discussed in the review in details. Next, please. But I would like to highlight that detailed studies of transporters can also lead to some unintended consequences. For example, studies of the transporter substrate specificities of a companion cell localized transporter OPT3, next please, have led to the establishment of the existence of the shoot-to-root copper signaling. For example, we showed that OPT3 transports copper in addition to iron, which was established quite some time ago, and loss of this function in the mutant leads to overaccumulation of copper in leaves, for example, as you can see, X-ray imaging of copper distribution. And then it really alters the shoot-to-root signaling in the mutant, and then that can be rescued by copper application. So this is really discussed in detail in the review, and we'll be happy to discuss after my talk as well. Next, please. Copper uptake strategies in grasses remain much less understood. In the review, we discuss existing evidence for the 3 not mutually exclusive scenarios. So first is that copper can be taken up as copper-1 in a reduced form. This implies the existence of the root surface reductase activity in grasses, which is analogous to Fro3/4 enzymes in Arabidopsis and other dicots. However, we raise the point that in grasses, for example, in rice, brachypodium, and corn, only 1 or 2 genes equivalent to Fros exist. So this strategy should be explored in more details. Another scenario is that copper can be taken up as in oxidized form as copper-2, and this would imply the existence of ZIP-like function or uptake via YSL-like transporters, which we showed can, in addition to transporting metal chelates, can also transport free copper ions. Another scenario is that copper can be taken as copper ligand complex, such as copper complexed with nicotianamine or nicotianine derivatives, phytosiderophores. So these are just still sort of suggestions and hypotheses based on some of the existing literature. But again, that has to be determined experimentally. The internal copper transport in grasses also involves members of YSL, HMA, and COPT family. However, our knowledge there is more limited compared to dicots. Next, please. Next, yes. So I also would like to highlight that studies of the Brachypodium phloem-localized copper transporter, YSL3, in addition to showing its role in source-to-sink copper partitioning, including seeds, have identified the role of copper in developmental of such an important agronomic traits, such as seed size and seed protein level. And so future studies should address the mechanistic basis of this phenomenon. Next, please. Even less is known about intracellular copper transport. Chloroplasts, which are main copper sinks in photosynthetic tissues, take up copper via P-type ATPases, HMA1, HMA6, and 8, as was shown in Marino Spillone and Daphne Sejan-Burney labs. Surprisingly, how copper enters mitochondria, the second cellular copper sink, is unknown. And so we hypothesize that one of the PHT3 family members, which are homologous to copper transporters from yeast and animals, might be involved. An important site of copper entry into the ER is AT-RUN1, alias HMA7. Initially, this transporter was discovered in Joe Ecker's lab and was shown to be important for copper delivery to ethylene receptors, which are also localized in the ER. Next, please. An important notion, which is discussed in the review and Uta highlighted in her talk as well, is that copper forms exceptionally stable complexes with organic ligands, as explained by the Eyring-Williams series. And so this high copper ligand stability can lead to mismethylation and protein inactivation. And one of the ways that cells prevent this from happening is by utilizing chaperones to ensure accurate methylation by protein-protein interactions and also ligand exchange reactions. And so in the review, I mean, quite a few chaperones have been identified in plants and yeast as well. And they're all, I mean, for the sake of time, I won't go through it. And I invite you to look into the review if you're interested in this topic. Next, please. The review, next, please, again. Yes, the review also discusses the mechanism that plants use to avoid deficiency and buildup of toxic levels. And this regulation is mainly attributed to the master regulator of copper homeostasis SPL7, identified in Shikanai lab. And SPL7 is a homolog of the copper sensor in Chlamydomonas, which was discovered and characterized in Sibia Merchant's lab. So SPL7 regulates the so-called copper economy metal switch mechanism, and that to prioritize copper allocation to essential cellular functions. This involves microRNA degradation of genes which encode abundant and functionally redundant copper proteins. Their depletion is compensated by the isoforms that utilize other metals and most commonly iron. SPL7 also collaborates with the transcription factor CETF1 to regulate copper uptake, delivery to shoots, including reproductive organs. Among SPL7 downstream targets is CETF2, which was recently identified in Uta's lab. However, we still need to understand better its contribution to copper homeostasis. Next, please. One of, as you know, this was a silver lining in some of the talks, and Uta also highlighted as well, is one of the fascinating questions in the field of metal homeostasis is how plants sense deficiency or abundance. So concerning copper, early insights on copper sensing came, well, very early insights came from studies in bacterial system, but regarding plants, the insights came from studies in Chlamydomonas in Sibia Merchant lab. So they identified copper response elements in the promoters of copper responsive genes. These copper response elements here in a figure shown Cura. And the copper response regulator CRR1 of Chlamydomonas and its homolog SPL7 in Arabidopsis and was also identified recently in rice recognize these motifs. Both CRR and SPL7 also contain zinc-binding SBP domain. And this SBP domain is regarded as a putative mental sensing region in CRR1. In addition, CRR1 has 4 putative metal binding and cysteine-rich motifs in the C-terminal. So these motifs, they're not conserved in SPL7. However, we did some search of the C-terminal of SPL7, and we found putative metal binding sites. Their contribution to metal binding still has to be explored. Please hit next, please. And next, please. So I really, for the sake of time, I would not go through all of the proposed mechanism of metal sensing. I do invite you to the review to look at it. But the idea is that actually under copper sufficiency, so zinc is needed for CRR and SPL7 binding to its targets. So under copper sufficiency, copper actually prevents the binding. Under copper deficiency, there is a possibility to make it happen. And so also some of the metals were shown, such as nickel for CRR and cadmium for SPL7, have been shown to sort of mimic copper and zinc in their recognition of copper responsive elements in the targets. Then let's please, next please. Yeah, thank you. Just, to summarize, I mean, there's quite a bit of knowledge already on how copper homeostasis is achieved, and it's increasing from year to year. And what I'm highlighting here, what probably one of my personal interest as well, but I think it's sort of you've heard it across different talks. I think we still lack complete understanding of the transporters which are involved in metal transport and distribution in plant tissues. We are lacking still, knowledge of the chaperones regarding copper that would bring, copper into different, proteins and transporters as well in a cell. We don't understand how copper is taken up in grasses. That needs attention because grasses, they encompass major cereal crops such as rice and wheat. We do not know how copper is imported to subcellular organelles, mitochondria. We don't know. It's second major copper sink, still unknown. Copper sensing and signaling is unknown, and broader physiological role of copper beyond its static role as a cofactor, similar to what was shown in animals, also needs to be explored. And with this, thank you for the attention.
Thank you very much, Olena. Thank you very much to all the speakers for this webinar. We are already very advanced in time. I've seen a question and answer box that several questions have been answered already. I invite all the attendees who have further answers to address directly the authors. The authors, I'm also an author, we are happy to answer any question that is left. So with this, I would like to thank you for attending this webinar, and thank you for the presentations again. And I'm going now to hand over again to the person from the publisher.
Thank you all. All right, great. I know we are over time, so as Ingo said, please do forward any questions that you do have. Before I let you all go, I do want to say a huge thank you to the speakers and to everyone who attended today. You can read the full articles and all the other great pieces in the journal over at cambridge.org at QPB. We will be posting the recording of this talk as well, so if you missed anything or are craving a rewatch, look out on the homepage or our social accounts for those links. But otherwise, thank you again so much and take care. Bye.
Thank you.